This presentation is about ketone metabolism during diabetic ketoacidosis. As an introduction, people who do not have diabetes produce insulin correctly in their pancreas. The insulin binds to insulin receptors on the surface of a cell, which opens up glucose channels. The glucose channels then enter the cell, where glucose is metabolized for energy. You can see some little animations there. When someone has diabetes mellitus type 1, the pancreas does not produce the insulin required to open up the glucose channels and allow glucose to enter the cell. This results in glucose building up outside of the cell and in the blood, which leads to high blood sugar. The lack of glucose in the blood will lead to fatty acids being metabolized into ketones. The buildup of ketones is what results in metabolic acidosis. Metabolic acidosis has severe side effects that can eventually lead to death. A person with diabetes mellitus type 1 will have an insulin deficiency. Insulin usually suppresses the glucagon hormone, but at low levels of insulin, glucagon levels increase. This increase in glucagon will stimulate gluconeogenesis and promote glycogenolysis while also inhibiting glycolysis. This results in hyperglycemia, or high blood sugar levels. Lipolysis is the metabolic process in which fats are broken down into glycerol and free fatty acids. One of the methods in which lipolysis occurs is through hydrolysis. ATGL, or adipose triglyceride lipase, hydrolyzes the first fatty acid group. Glucagon stimulates this path and insulin inhibits it. Since insulin levels are low, there is plenty of glucagon to stimulate this path. HSL, or hormone-sensitive lipase, hydrolyzes the second fatty acid group and acts as a rate-limiting enzyme. Glucagon also stimulates this path. MGL, or monoacylglycerol lipase, will hydrolyze the monoglyceride into a fatty acid and glycerol group, once again being stimulated by glucagon. Here is the overall lipolysis reaction resulting in 3 free fatty acids. The 3 free fatty acids, or FFAs, are taken to the mitochondria to begin beta oxidation. The FFAs must first be converted to a high-energy acyl coenzyme A before entering the mitochondrial matrix. This conversion happens in the outer mitochondrial membrane. This is the pathway for the beta oxidation of the fatty acyl coenzyme A, which produces the acetyl coenzyme A. FAD oxidizes the acyl coenzyme A into enoyl coenzyme A. Enoyl coenzyme A is then hydrated into a hydroxyl acyl coenzyme A. The hydroxyl acyl coenzyme A is oxidized into a ketone by NAD+. This ketone is called beta-ketoacyl coenzyme A. Beta-ketoacyl coenzyme A undergoes cleavage to form acetyl coenzyme A and a shortened fatty acyl coenzyme A. This shortened fatty acyl coenzyme A will continue to go through this cycle multiple times. Here is the overall beta oxidation reaction that produces acetyl coenzyme A. In a healthy body, acetyl coenzyme A is diverted to the TCA cycle to produce ATP. During diabetic ketoacidosis, oxaloacetate is diverted to gluconeogenesis, which depletes it. With low levels of oxaloacetate and high levels of FFAs, there is an excess of acetyl coenzyme A. The acetyl coenzyme A is converted into ketone bodies to be used as an energy source due to the lack of glucose. This is the process in which acetyl CoA is converted into ketone bodies. Two molecules of acetyl CoA undergo a condensation reaction, meaning that the two molecules are combined into one. Naturally, the availability of acetyl CoA limits this reaction. The product is acetoacetyl CoA. Acetoacetyl CoA is reacted with HMG-CoA synthase and then reacted with HMG-CoA lyase to form these ketone bodies. The dominant ketone body formed in DKA is beta-hydroxybutyrate. Acetoacetate is also formed in DKA, and it undergoes a spontaneous reaction converting it into acetone. Acetone is volatile while in the lungs and causes patients suffering from DKA to have fruity breath. The availability of FFAs, or free fatty acids, as well as levels of glucagon, which is increased due to insulin deficiency, limit how many ketone bodies are produced. Ketone bodies are acidic. While they can be used for energy, high concentrations cause widespread acidosis throughout the body. For our primary literature discussion, we chose a study based off of acetone metabolism during DKA. The study examined acetone metabolism in DKA patients. The plasma acetone concentration in the patients ranged from 1.55 to 8.91 micromole, and it often exceeded acetoacetate levels. Acetoacetate is shown in the figure as ACAC. The average acetone production rate was 265 micromoles per minute per 1.73 meters squared, accounting for approximately 52% of the estimated acetoacetate production rate. A positive linear relationship is observed between the plasma acetone concentration and the percentage of acetone excreted via breath, a reminder that acetone in the lungs makes a patient's breath smell fruity. In the figure, black arrows at the top indicate at what hour insulin was administered. You can see the reduction in the ketone bodies when insulin is administered. It is also worth noting that the beta-OHB is the highest concentration of ketone body, with ACAC at second and acetone at third. Acetone is noticeable in the breath, but it is not what you want to measure for when diagnosing DKA. In conclusion, DKA is a serious threat. Insulin administration and IV fluids can save the lives of patients. The primary literature cited showed that insulin administration is an effective tool in treating DKA. Having an understanding of ketone metabolism can help us appreciate the metabolic shift during insulin deficiency, understand the fact that the liver plays a central role in energy adaptation, and that the acidity of ketones is a double-edged sword in the fact that it can provide fuel for our bodies, but it also poses a serious threat. Some clinical takeaways of this presentation is the fact that BHB is the main ketone produced in diabetic ketoacidosis. This is what you want to measure for a diagnosis, not acetone. Though the fruity smell of volatile acetone can be used to assist in a diagnosis. Here is some supplemental reading to further elaborate on the topics of this presentation, as well as the references used to acquire the information.